Good morning students, welcome to LEELA'S tutorial. So today we will discuss about marker assisted selection. So this is one of the marker identifying technique which is indirect. So in previous classes we have discussed about the molecular markers, which are the small DNA fragments or the molecules which are used to differentiate or identify a particular organism from a pool of DNAs. Right? So here we have discussed about first generation markers, second generation markers and third generation or new generation markers. So let us discuss about this marker assisted selection, which is also referred as mass selection. Right? So mass selection is an indirect selection of better performing breeding individuals. So already in the livestock we have discussed that when you are want to produce a good environment for the livestock, you have to maintain the proper feed, then environmental conditions, then the hygiene. So apart from these, you are producing a better quality livestock by transgenesis, otherwise by means of in vitro fertilization. So the characters present in these animals, or the superior characters or the superior characteristics, they must retain for decades, but not for only 1 or 2 generations. So as the crossbreeding occurs, there may be the dominant genes may be lost gradually. So in order to avoid that from the livestock, you are going to select them by using this mass selection process, and you are going to develop your farm by only having your better performing breeding animals. So here, what do you mean by better performing breeding individuals? That means they must be free from any genetic diseases or genetic disorders, disease resistant, antibiotic resistant, and they must produce a good or improved quality of the products, and desired behavior characteristics are observed. So all these characters, see here, these can be observed normally the phenotypically, right? But these disease resistant and antibiotic resistant, everything must be evaluated by checking the genotype of the organism, right? When you have an progeny, you can differentiate, or you can say that they all are healthy and they are better performing by seeing at the phenotype. But genotypically they may not receive those dominant or superior genes, right? So in order to check, or in order to select the better performing breeds, we have to go for the mass selection process. So here the mass selection is based on traits when the phenotype is not easy to evaluate the better characteristics of animal variants, right? So we have to go for the genome analysis. So when you go for the gene typing or genome analysis, here we are making use of certain markers, right? We make use of certain markers in order to check that superior quality genes are inherited to the progeny or not. So let us see one example. So here I have taken the mature bull, right? This bull is having an superior qualities in their genes. So here you can see it is having an dominant A and the recessive A. So this is responsible for the superior quality of that bull. The better performance of the bull is because of this genes present, right? So here I am selecting A as a marker. So A acts as a marker for this selection process. See, now you have the female animal which is having both the recessive genes, right? So here I have tagged a marker only to the male one because I want these super qualities must be reflected in the progeny, right? So when these two are crossed, or here we go for the artificial process where the semen is collected and sperms are separated. Then from the cow, the super ovulation is given so that it will produce multiple ovules. That is called as multiple ovulation process. So that instead of producing one ovule, it will be producing 4 or more than that. So here 4 or up to 6, right? So in multiple ovulation process, then after this you are going to extract those ovules, right? So here during that process, right? There may be some damage. The damaged ovules will be rejected, right? Ovules or the oocytes. Then you are going to take the healthy oocytes, right? To those healthy oocytes, the sperm cells are incubated, right? So that the fertilization occurs. So artificial fertilization occurs in the lab. So once the embryos or ovulation, after the ovulation, when the fertilization is completed with the sperm, here they develop into an embryo. So these embryos start developing. So then after this, the developed embryos at the blastocyst stage, they are introduced into the surrogate mother. So you are not going to introduce all 4 embryos into single surrogate mother. Instead, you are selecting 4 surrogate mothers and introducing each embryo into each surrogate animal. So now after the gestation period, that is after the time of introducing to the birth, right? After the gestation period, you can see the surrogate animals has produced 4 surrogate animals produced 4 calves. So here you can see the first calf is not having that marker gene, right? But second is having that capital A, and third one is not having, and fourth is not having. But phenotypically all calves seem to be healthy, right? So here you want to differentiate this one or select this one. So what you do? You go for the progeny testing. So in the progeny testing, you will be selecting the progeny or the calves with the gene marker A, which was present in the parent, male parent, bull. Right? So after progeny testing, here you can separate out of all 4 cows, you have separated this second cow as it is having capital A, the marker. So here it is pre-selection of marker A in the calves or marker A in the progeny. Right? So out of all progeny, only 1 calf was having this superior quality genes. So now these calves are grown into a complete matured animal or the bull, and this is referred as mass assisted selection bull. Right? So now this bull will have all the superior qualities which are present in this male parent. Right? So this is an indirect method of selecting or using the markers as in order to produce a better performing breeding animals. Is that clear? If you have any doubts, you can ask me. Subscribe for further videos.